Hello, my name's Sandra Wilkes and I'm a lecturer in medical microbiology at the University of Southampton. First of all, I'd like to thank the organisers for inviting me to talk today, and I'm really excited to be able to tell you about some of the work that we've been doing on a quite large programme of work looking at the role of biofilms in indwelling catheters and using some advanced techniques to help us understand the problem of biofilms and also how we can use this knowledge to improve our control strategies and treatments. So first of all, what are biofilms and why are we interested in them? Well, biofilms are formed by groups of bacteria which attach to a surface or an interface. So let's think of a surface in this case. So one of the easiest ones is if you run your tongue over the surface of your teeth, they can feel a bit rough, and plaque is a biofilm. So biofilms are around us. And where we have these bacteria, these cells, sticking onto a surface, they start to form a 3D structure, and they form microcolonies and build up into almost like a little city of bacteria, where you have these 3D structures of groups of bacteria surrounded by channels carrying water and nutrients around, so almost like a transport system. And the bacteria are held together in a matrix, a kind of a glue or a scaffold, and that's something that they produce themselves. It's called extracellular polymeric substances, or EPS, and this can be composed of all sorts of different compounds, including carbohydrates, proteins, lipids, and even extracellular DNA. And it's really important that we consider biofilms differently than when we consider bacteria in just a liquid or a planktonic phase, because the biofilm itself, the structure, can give them tolerance and resistance to different types of chemical and mechanical attack. So, for example, the action of antibiotics, you need a much higher concentration to actually attack a cell within that biofilm state. And also they can act as a reservoir for pathogens. So you can have a biofilm forming and just a small number of pathogenic bacteria, which will almost like hide out within that structure. And that protects them from any kind of chemical or mechanical attack. So why are we interested about biofilms when it comes to urinary catheters? Well, we've known for a long time that they can develop on either the external or the internal lumen of the catheter. And the presence of biofilms can contribute seriously to the frequency and prevalence of catheter-associated UTIs and cause catheter blockages. And these infections and blockages can often reoccur, especially in long-term users. Previous studies have shown that around 50% of patients undergoing long-term catheterization will experience these crystalline encrustations that lead to blockage. And on the left here, we have an image showing a completely blocked lumen of a catheter. When we're thinking of blockages, these are most commonly due to the presence of a bacterium called Proteus mirabilis, which produces urease, causing the pH of the urine to increase rapidly. And this causes crystals to form and these really dense encrustations which go on to block the catheter. So it's really a serious problem. And also, if we look back at the increased incidence of UTIs, we also find that in many UTIs, we get evidence of multi-drug resistant strains. And the urinary environment has been the initial place where many of the currently known multi-drug resistant strains have been first isolated. So what are we doing? Well, we're using an interdisciplinary approach to take what we know about microbiology in the lab and improve our testing and our modelling to try and make this more appropriate for real-world comparisons. And by doing this, sort of get more robust data before going into patient trials. And we're using particularly, and what I'm going to talk about today, is very advanced non-destructive imaging techniques to really help us understand the development of biofilms, and then use this information to help us identify new antibiofilm materials or strategies. So what do we mean by improving laboratory testing? Well, it's really important that we replicate the conditions that actually exist. So instead of using just normal nutrient media, we're using a physiologically correct artificial urine medium because we know that biofilms develop in different ways under different conditions. And this can have a really profound effect on the results you get. We're also making sure that we use flow models, such as the existing bladder model, to replicate the action of catheters in situ. However, we also know that there are limitations with this design. So we are working with experts in flow dynamics and microfluidics to improve the design of this bladder model and eventually come up with a test system which replicates the whole of the urinary system. And then we can use this to look at the impact and input of new catheter designs. So this is very much a collaborative piece of work with microbiologists working with engineers and clinicians. So I mentioned that we were using some advanced imaging techniques. The first one that I'm going to describe is the use of EDIC or EF microscopy. It's got a long name, episcopic differential interference contrast and epifluorescence microscopy. And basically, to simplify this, what it allows us to do is to look at high magnification directly on surfaces that can be solid or opaque. And by using this system, we can look at the catheters directly with only very minimal sample disruption. So we don't have to remove the biofilm. We can look at the biofilm in situ on the catheter surface. What are the kind of results we get? Well, even just looking at clean, unused catheter surfaces reveals a lot of information. So in this EDIC image, on the left-hand side, we have an uncoated silicone catheter, and on the right-hand side, we have a hydrogel latex catheter. And neither of them are really very smooth, especially the hydrogel latex. We've got a very rough, disordered surface, which provides many attachment sites for bacteria, increasing that biofilm susceptibility. On the silicone catheter, it does look smoother, but you can still see that it is rough. We've got striations running the length of the catheter from the manufacturing process, and lots of pits and crevices, again, offering attachment sites for bacteria and subsequent biofilm development. What do the biofilms look like? Well, these are some example images from E. coli, which is not a very strong biofilm forming bacterium. But here we have the EDIC image, and you can just about see some darker areas. When we stain these with a staining system that stains live bacteria green and dead bacteria red, we can see that the very dark areas match where the bacteria are. And these are these 3D structures that I was talking about forming the biofilm. And by merging the images, we can start to see how those bacteria are interacting with the material surface. We found that this happens very quickly. If we look at exposing catheters to Pseudomonas aeruginosa, which is a very active biofilm forming bacterium, within 6 hours of exposure in artificial urine, almost the entire catheter surface has been covered with just some very small channels running in between, these nutrient transport channels. After 24 hours, we see even more coverage of the entire catheter surface. But an interesting event happens after 48 hours, where we've got a far more patchy biofilm forming. The biofilm itself is now more 3D, but it has more of a mosaic form, and you can start seeing just about the bright areas where we're seeing the catheter surface again. And this is where we have this dynamic life cycle of the biofilm, where we have cells attach, they develop, and then eventually they lift away, either just by parts detaching and floating away in the urine, or we have active dispersal. So the biofilm actively releasing bacteria to go and colonize other areas. So it's a very complex system, and it needs to be thought of as a system. Thinking of Proteus mirabilis and the causative agent of those encrustations and catheter blockage, a study we did a few years ago really helped us track how these blockages happen. So here we have some EDIC images. On the left-hand side, this is after only 1 hour exposure, and we've got just individual cells attaching to the silicone catheter surface. However, after just 2 hours, it looks like there's a lot more material there. There's a lot more coverage of the surface itself. And if we use some specific stains to look at what's there, what we actually find is here we're using a blue stain to label bacteria, and we've got some individual cells labeled, but then we're using a general sugar residue stain that causes red fluorescence. And that's what the majority of this material is. And this is called a conditioning film. It's caused through the action, the presence of the bacteria being there, but it's coating the whole of the catheter surface, and it's creating more attachment sites. It's making it easier for a biofilm to form. So it's a really important component. If we continue mapping this across time, we find that we get even more development, and we can see key points in the crystalline biofilm formation. So after 3 days, we have complete coverage of the catheter itself, but then we get this very fine microcrystalline sheet which sits on top of this initial covering of the catheter. After only 4 days, we suddenly get a huge amount of diffuse crystalline material forming. This is apatite crystals coming first. And the pH at this stage has gone up to around pH 9 or even higher. After 5 days, we then get the formation of really distinct struvite crystals, again embedded within this diffuse crystalline mass and attached to these lower layers of completely covering the catheter. Using our microscopy system, we can then take composite images. So all of these are composites of around 100 to 200 very thin slices through crystals. And what we see here, in this case, we have a single struvite crystal, and it's completely colonized with bacteria. So the crystals themselves become another surface for colonization. So rather than thinking we have bacteria, pH rises, crystals form, that's the blockage, what we're actually finding is the whole system is interlinked, and it's very dynamic, and it all works together. And from this study, we've been able to identify 4 main stages in crystalline biofilm development before we get to that point of having complete blockage. And this is helpful because we can then look at control strategies to target each of those stages and focus on where would make the best intervention. We've also taken this a stage further and used volume imaging. So in this case, we're using a microcomputed tomography imaging to look at the 3D structure and really understand the interaction between the cells, that biofilm formation, and the surface itself. Here we have some synchrotron images showing how that very early biofilm development really attaches very strongly to the catheter surface. And if we take the model further, we can then look at how the entire lumen is blocked. And this is really useful because then using the micro CT scans, we can see how these crystals are forming together and come up with video images like this, which really show how the whole catheter is being blocked. And then using this information and putting it into flow dynamic studies, we can look at whether there's key areas of the catheter that are susceptible to blockage. So I mentioned that we're using these advanced understanding techniques to help us understand, to look at control approaches. And I really want to talk about two examples. So if we think about how we can control the development or prevent the development of biofilms, we can think about having impregnated or coated materials, and these can include antibiotics. There are catheters available that are impregnated with antibiotics or coated with antibiotics, or they can be other antimicrobial agents, such as the silver catheter that we're all familiar with. We can also look at whether we can apply mechanical methods to dislodge biofilms or prevent them forming. So can we increase shear flow to move that biofilm to sort of shake it off the surface if we're looking at the lumen? And I just want to talk about two studies we've done. The first one looking at different catheter materials and focusing on the use of silver, and the second one looking at the use of low frequency surface acoustic waves in the UroShield device. So a bit of example data from our catheter material study. This is about to be published. We looked at E. coli, Pseudomonas aeruginosa, and Proteus mirabilis, and we tested 3 different catheter types: silicone, hydrogel latex, and silver alloy hydrogel latex. And we looked at tracking biofilm development over time, and we did 3 different measurements. We calculated the number of very actively growing cells, the culturable cells. Then by direct microscopy, we assessed how many total cells there were, so live and dead, and how many dead only. And really the take-home messages from this, in these 3 graphs, we have silicone, hydrogel latex, and then our silver. Really, the first take-home message from this is that there's very little difference between all 3. The silver is certainly not showing an increased activity in preventing biofilm formation. And that's very much, this is over 72 hours, that's very much what has been found in clinical trials now. What we are seeing is that we're getting development of a sort of a static population. So with silver in particular, we find that we are getting an increased number of bacteria which are not actively growing, but are still alive and are still there. So they are still potentially able to infect, potentially able to cause an infection, but they're going undetected. So in this case, at best, the material is being bacteriostatic rather than bactericidal. And the implications for this, from a clinical sense, is really unknown. And that's a really interesting area of work that we're now focusing on. I mentioned that we're also very interested in looking at mechanical methods. And this is a study that we've been carrying out with a company who have a device called the UroShield. We are part of the National Biofilms Innovation Centre, which is based at Southampton. And this helps us as academics work directly with companies and give them access to our advanced analysis methods in their product development or product testing. And this is one of those projects. And in this case, the UroShield device is a small actuator that gets clipped to the external surface of the catheter, and very low frequency surface acoustic waves are propagated through to that device and then travel the length of the catheter, affecting both the inner and outer surface. The company have had very positive results and feedback from patients, but they asked us to do a laboratory model study using our bladder model setups to try and understand what was happening to the bacteria. And to give an example of some of the data, what we're showing here is the percentage reduction in colonization compared to an untreated control. So if we get 100%, we've removed all the biofilm, or the action of the UroShield has removed all the biofilm. What we found was after 48 hours, looking at different sections of the catheter, so we have the tip in the bladder, midway, and then the leg bag end. When we look at a clinical urinary strain of E. coli, we're getting a near enough 100% reduction in biofilm formation, which is very positive. For Proteus, we find it's a little more variable. So at the catheter tip, we're getting, again, an almost 100% reduction, but this is reducing to around just over 50% at the leg bag end. But it's really important to see what these results mean, because even if we could only reduce the formation of crystals and encrustations around the catheter eye holes, that would be a great improvement for people who are getting regularly blocked catheters. So what have we found overall from this work? Well, I'm just giving you a very small snapshot of the kind of studies that we have underway. But what we do know is that bacteria will attach to the catheter surface extremely rapidly. We can start seeing attachment in less than 15 minutes. And after only 2 hours, they're surrounded by this polysaccharide-based material, this conditioning film, which only increases the chance of biofilm development. So maybe we should be targeting that rather than the bacteria themselves. We know that if we're looking at catheter blockage, that these really complex crystalline biofilms develop very quickly, and they really are an integral part of the biofilm structure itself. So again, a bit of a different understanding on how they form. And by using our advanced imaging techniques, we're getting so much more knowledge on how these biofilms develop. They're really helping us track and understand these different stages of development, which helps us pinpoint sort of targeted areas that we can focus on. And this can only help us with understanding how different material surfaces, different surface structures and characteristics influence biofilm formation. How does this all affect our control strategies, our treatment options? Well, the more we can do robust and appropriate laboratory testing, it's only going to help us in the development of new materials and new catheter designs. And we've shown from this work that we need to be really careful that we're doing appropriate testing, especially when we're looking at new antimicrobial materials. We can have increased production of these static populations, these viable but non-culturable, non-actively growing cells, which can actually still be infective and cause a problem. We've also shown that mechanical methods can be very useful, but they also need careful testing and validation. And we can do more of this in the laboratory before going into patient trials. And working together in an interdisciplinary group can really help us understand all those influencing factors, and we hope in the future improve catheter design and management. And with that, I'd like to give a few acknowledgements to my colleagues, in particular Professor Mandy Fader, who leads the Bladder and Bowel Management Group in the School of Health Sciences, and all the other members of that group. We work very closely as a group, and also Dario and Orestes, who helped with all the micro CT work. The projects I've shown today have been funded by the Institute for Life Sciences and the National Biofilms Innovation Centre, and we're really grateful for all of their continued support. And at that point, I will just leave you with a final video as we travel down through one of these complex crystalline biofilms, showing a struvite covered in colonizing Proteus mirabilis, and then surrounded by this large amount of diffuse crystals, really complex, dynamic community. Thank you very much for your attention, and I'd be very happy to take any questions.